Hello, welcome to this presentation. I am S.O Piennes. I'm the president of the medical students of Osun State University, Oshogbo campus, Osun State, Nigeria. I want to give this presentation, which is basically on GIT physiology. This presentation will help you in two ways. Number one is to give you quick revision of basic things that you need to know in GIT. Then secondly, it will help you to have an overview of what GIT is all about. So I implore you to follow as we start. Now let's have the highlights of the presentation. Number one, we start with introduction. Number two, we look at glands in the alimentary canal and those related to it. Number three, we look at the primary alimentary organs. Number four, we look at accessory or secondary alimentary organs. Number five, we look at GI movements. Number six, we look at GI potentials. Number seven, we look at GI hormones. Number eight, we look at digestion, absorption and metabolism of carbohydrates. Number 9, we look at digestion, absorption and metabolism of proteins. Then number 10, we look at digestion, absorption and metabolism of lipids. Now all these will be done within 30 minutes. So let's start. Now starting with the introduction. GIT physiology, that we call gastrointestinal physiology, is a physiology that deals with the digestion, the absorption, and the metabolism of food stuff that we take in. And we need to understand that when we talk about GIT, we are not restricted to the stomach and the intestines alone, but we also consider the alimentary canal as a whole. Okay, now looking at the alimentary canal, it extends from the mouth to the anus, which is about 10 meters at stretch. Now we can have an idea of what this is all about. Now we have injection, secretion, fragmentation, peristalsis, digestion, absorption and defecation. Now all these processes take place along these tracts, the alimentary canal. Alright, now let's consider the wall of GIT. The wall of GIT, that is when you make a transverse section of it, what you can see. Now we have four major layers. We have the mucous layer, we have the submucosal layer, we have the muscular layer, and we have the adventitial layer. The mucosal layer consists of three parts. We have the epithelial lining, that's the immediate part that's close to the lumen. So every surface are lined by epithelium. That's what we learned from histology. Then epithelial lining followed by lamina propria, then followed by the muscularis mucosae. All those ones that compose the mucosal layer. The submucosal layer consists of some loose connective tissues, like blood vessels, nerve fibers, and some connective fibers. Alright. Then the muscular layer consists of the inner circular layer and outer longitudinal layer. But in the stomach, we have another layer there that we call inner oblique layer. So the stomach has three layers, three muscular layers: the inner oblique, the middle circular, and the outer longitudinal layers. Okay. Then the fourth layer is the adventitial layer, which can be serosa or fibrosa. That is, it can be serous membrane or fibrous membrane depending on the parts that you're talking about. That is in the GIT. Alright. Now let's go to the second part, which is glands of the alimentary tracts. The glands of the alimentary tracts. Now let's take it regionally. Let's start with the oral region. In the oral region, we have the salivary glands. The salivary glands are major and minor. We have major salivary glands, we have minor salivary glands. Okay, the major ones are the parotid, the submandibular, and the sublingual. The parotid is majorly serous. Okay, the submandibular and the sublingual, they are mixed. Okay, so we also have the minor salivary glands. There's so many, but the major one is von Ebner's gland, which is majorly mucous secretion. We have some other minor ones like palatine glands, like labial glands, lingual glands, and so on and so forth. That is the glands in the oral region. Let's go to gastric region. In the gastric region, we have the gastric glands, there are pylori glands, fundy glands. The gastric glands consist of a number of cells that's release different secretions. We have surface mucous cells, we have neck mucous cells, we have parietal cells, we have the chief cells. The parietal cells secretes HCl and intrinsic factor, while the chief cells secretes pepsinogen and gastric lipase. Okay, so let's go to intestinal region. In the intestinal region, we have a number of glands there. Now we have the small intestinal glands that release sulcus and entericals. We have the exocrine pancreatic glands. Okay, we have the hepatic gland that is in the liver now, because the liver is a gland actually that secrete bile. Okay, we have the large intestinal glands. That is a function of some lining cells, goblet cells that release mucus majorly. Okay, now please note that the cells that line the surface of the intestines are called the enterocytes. Okay, enthero, that's inside, internal enterocytes. All right, now let's quickly talk about the regulation of these secretions. The regulation of these secretions is governed by two major control. That's nervous and hormonal control. Sometimes it can be both. All right, let's go to the third part. That's looking at the primary alimentary organs. The primary alimentary organs are the mouth, the oral cavity, the pharynx, the esophagus, the stomach, the small intestine, the large intestine, the rectum, the anal canal, and the anus. Now you realize that this ends at two openings. That's the mouth and the anus. Now mouth is an opening as well as the anus. Okay, so let's look at the secondary alimentary organs. The secondary alimentary organs or accessory alimentary organs, they have accessory function. They are not directly involved, but they have accessory functions. In the oral region, we have the teeth, the tongue, and the salivary glands. Those are accessory organs, in the oral region. In the abdominal region, we have the exocrine pancreas, we have the liver, and the gallbladder. All right, let's talk about the GI movements. The GI movements are basically contraction and relaxation. Okay, so when we go from oral region to distal parts, we talk about mastication, that is chewing. Talk about deglutition, that is swallowing. We talk about stomach movements, which are basically the peristalsis, the receptive relaxation, and hunger contractions. Okay, then from there we go to small intestines. Small intestinal movements, we have a number of them. We have mixing movements, we have propulsive movements, we have peristalsis movement too. Okay, then in the large intestine we also have movement too. We have segmentation, contraction, and mass peristalsis. That is in massive movement of feces. That is in the large intestine. Then we also have defecation. Defecation is a movement too that involves the emptying of the intestine. That is the distal part of the large intestine. Now let's talk about some reflexes: deglutition reflex and defecation reflex. Deglutition reflex is a reflex that causes swallowing. All right, we have three phases. We have the oral phase, pharyngeal phase, and esophageal phase. The last two phases are involuntary, while the first phase is voluntary. All right, defecation reflex, a reflex that triggers defecation or emptying of the rectum. Okay, now but we also need to note that we have some other reflexes that are involved in gastrointestinal movements. We have gastrocolic reflex, we have gastroiliac reflex, we have ileocolic reflex. Okay, so two innervations actually mediates defecation reflex. Number one is intrinsic defecation reflex and extrinsic defecation reflex. Intrinsic defecation reflex is mediated by the enteric nervous system, okay, which is the intrinsic innervation of the GIT. The extrinsic defecation reflex is mediated by the autonomic nervous system, which is the parasympathetic part of it. Now we need to note that the extrinsic defecation reflex is stronger and acts most of the time, Now let's talk about the reflex proper. The stimulus is the stretch receptors in the rectum become activated by droplets of feces from the distal part of the large intestine. So the moment the stretch receptors are stimulated, then they send the impulse via the afferent nerve, which is the pelvic nerve fibers, okay, to the integrating center. Now I'm majorly talking about the extrinsic defecation reflex. Okay, so the integrating center is the S2 to S4 sacral segments of the spinal cord for extrinsic parasympathetic nerve fibers. The enteric nervous system, okay, and the enteric nervous system for intrinsic. The efferent nerve is actually parasympathetic fibers in the pelvic nerve. Then next to the effector, the parasympathetic fibers innervate the effector, which are majorly the internal anal sphincter, the pelvic diaphragm muscles, the abdominal muscles, and some other muscles that are needed for defecation act. All right. Now, finally, the response is the arrival of feces in the rectum and the anal canal, and so you feel defecation. Feel that you want to defecate, ready for evacuation. All right. So let's go to GI potentials. The GI potentials are form of potential that are majorly slow waves and spike potential. The slow waves are caused by interstitial cells of cardia. Those are pacemaker cells in the GIT. The slow waves, otherwise known as the basal electrical rhythm, give rise to spike potentials, which rise beyond the basal level. Which, when rise beyond the basal level, give rise to spike potential. The slow waves actually give rise to spike potential when it rise beyond the basal level. All right. Now the spike potentials are the ones that cause smooth muscle contraction and not the slow waves. So please note that. Then the extracellular calcium ions are actually required for adequate tonic contraction of the smooth muscles. The smooth muscles in the GIT are actually unitary type of smooth muscle. Okay, so now let's have a short break. I'll be back after this timeout. Relax. So welcome back. Now, next to the seventh topic, which is the GI hormones. Now we have a number of hormones that are secreted along the GIT. Now let's start with gastrin. Now I'll be talking about their primary action, even though you have a number of secondary functions too that they perform. But when you know the primary action, it helps you to know other functions that they perform. All right. So talking about gastrin is released in the stomach, just like the name, gastrin. Stimulates gastric secretion and motility. That's the primary action. So when it does that, it stimulates other functions like the growth of the gastric mucosa, the gastric contraction, the digestion process, and things like that. Okay. Now the next hormone is secretin. Secretin, just like the word secretin, causes profuse secretion. Profuse secretion of exocrine pancreas. Okay, that's contain more of watery enzymes and bicarbonates. All right. Now the next hormone is CCK. That is cholecystokinin. Cholecystokinin stimulates the cholecyst contraction. Cholecyst is a gallbladder. Chol means by, cyst means gall or a small cavity. So cholecystokinin stimulates the contraction of the gallbladder, thereby causing the release of bile. All right. Now the next hormone is motilin. Motilin, just like what motilin causes motility. So it stimulates motility of the intestines. Okay. Motilin is actually released profusely when you are fasting. So when you experience some contractions in your belly when you're fasting, that's the function of motilin. All right. Now the next hormone is VIP. That is vasoactive intestinal polypeptide. Vasoactive, so it's active on vessels. All right, active on blood vessels. So it increases the blood flow to glands, thereby increasing their secretions. So the primary action is to increase blood flow to glands, but secondary action will be to increase their secretion, because the moment there's increase in blood flow to glands, then they will be increasing their secretions. All right. So the next hormone is GIP. That is gastric inhibitory peptide. Okay, gastric inhibitory peptide, or glucose dependent insulinotropic hormone. That is all those ones they depict its function and its location too. It inhibits gastric secretion and motility, so it has inhibitory functions. The next hormone is peptide YY. Peptide YY. It has inhibitory functions, all true. Inhibitory functions to the stomach, to the pancreas, to the intestines. Okay. It also decreases food intake, so it decreases your appetite. All right. Neuropeptide Y is actually opposite of peptide YY. Neuropeptide Y has excitatory functions. It increases food intake. It increases blood flow to the intestines. Okay. Ghrelin. Ghrelin is an appetizing hormone. All right, released in the stomach. It increases food intake and appetite. Substance P increases the movement of small intestine. Okay, somatostatin has inhibitory functions, all true. Inhibitory functions to the stomach, to the pancreas, to the growth hormone. You remember somatostatin, that is growth hormone, inhibitory hormone, from the hypothalamus. Okay, it's still the same somatostatin. It's secreted in a number of areas in the body. Okay, it also inhibits the gastrointestinal hormones too, so it has inhibitory functions, all true. Pancreatic polypeptide, they increase glucagon secretion. Okay, glucagon and glycine both actually increase the blood sugar level. All right, so those are the major hormones, but we have other hormones too, other GI hormones that are released along the tract. We have bombesin, we have serotonin, we have neurotensin, we have enkephalins, we have dynorphins. Those are what they call them, opioid hormones. We have urogastron, we have enterocrinins, etc. Just like that. Then let's go to the 8th topic. That is the digestion, absorption, and metabolism of carbohydrates. Digestion, absorption and metabolism of carbohydrates. But to make it more easier, we're going to combine these three major classes of food: the carbohydrate, the protein, and lipids. So we start off with their digestion, then we go to their absorption, then we go to their metabolism. So for digestion of these three main macromolecules, carbohydrates, proteins, and lipids, now let's start. For carbohydrates, in the oral region, we have salivary amylase. Salivary amylase acts on polysaccharide and convert to dextrin and sometimes maltoses. Okay, so in the oral region, we have salivary amylase acting on carbohydrates. In the gastric region, we have gastric amylase. That one's very weak. Okay, so sometimes it may not act on the carbohydrate substrates. Then in the intestine, that's where you have a number of enzymes, that do the final job. In the pancreas, we have the pancreatic amylase. Okay, then in the small intestine, we have a number of disaccharidases. We have sucrase, we have maltase, we have lactase, we have dextrinase, we have trehalase, we have isomaltase, all to actually cause the release of monosaccharides. So polysaccharides are broken down to monosaccharides, like mainly glucose, sometimes galactose and fructose. Okay, so these are the products of carbohydrates. All right, now for proteins, in the oral region, there's no proteolytic enzyme. Okay, in the gastric region, we have pepsin from pepsinogen released by chief cells. Okay, now pepsin acts on protein to convert it to proteoses, peptones, and peptides. So protein is broken down to smaller units. Then when we get to the intestinal region, from the pancreas, we have a number of enzymes there. We have trypsin, we have chymotrypsin, we have carboxypeptidases A and B. Carboxypeptidases, we break down peptides at the carboxy end as different from aminopeptidases, which we break down peptides at the amino end, amino terminal. Please note that. Okay, that from the pancreas, we have trypsin, chymotrypsin, and carboxypeptidases A and B. Now from the sulcus and entericus, that's the intestinal secretion, we have dipeptidases, we have tripeptidases, and aminopeptidases. So please note that aminopeptidases is released by the small intestine, while carboxypeptidases are released by the pancreas, exocrine pancreas. Please note that. So the products are the amino acids and sometimes dipeptides, but majorly amino acids. Now for lipids, in the oral region, we have the lingual lipase. The lingual lipase acts on triglyceride, or triacylglycerol. Okay. Then in the gastric region, we have the gastric lipase. The gastric lipase is a weak lipase. Okay. Then when we get to intestinal region, so we have a number of enzymes there. From the pancreas, exocrine pancreas, we have pancreatic lipase, which is very active. We have cholesterol ester hydrolase or cholesterol esterase. We have phospholipase A and B. Okay, those are the enzymes from the pancreas. Then from the sulcus and entericus, we have intestinal lipases. All right, so they eventually cause the release of fatty acids, glycerol, free cholesterol, monoglycerides, and sometimes lysophospholipids. Okay, so these are the products of lipids. Now we've ended the digestion of the three major macromolecules, which are the carbohydrates, the proteins, and the lipids. Now let's go to the absorption. How are they actually absorbed? Now for carbohydrates, we'll be looking at two parts: absorption across the luminal membrane, absorption across the basal membrane. Okay, so movement across the luminal membrane, movement across the basal membrane. All right, so starting with carbohydrates, we said the major product is glucose, but sometimes you can have galactose and fructose. So for glucose, movement across the luminal membrane, we have sodium dependent glucose co-transport. That is a form of active transport. Okay, sodium dependent glucose co-transport, that is secondary active transport. Then the movement across the basal membrane, that is glucose, its movement across the basal membrane requires the glucose 2 transporter. Remember, we have a number of glucose transporters from 1 to about 6 or 7. Okay, so glucose 2 transporter is the one that is involved in the transport of glucose across the basal membrane. That one is a passive facilitated diffusion. Okay, so it's passive. For galactose, it's the same thing as glucose. For fructose, movement across the luminal membrane, we have glucose 5 transporter, glucose 5 transporter. That one is also facilitated diffusion. That's how fructose move across the luminal membrane with the aid of GLUT5 transporter. Now, movement across the basal membrane is via the GLUT2 transporter too. Okay, now let's go to the protein absorption of protein products, which is majorly amino acids. Amino acids are in two forms. We have L form and D form. The L form is via the sodium co-transport, just like the glucose too. Okay, sodium co-transport, that is secondary active transport, that is across the luminal membrane. The movement across the basal membrane is diffusion across the basal membrane to the blood. But for the D form of amino acids, the D form pass through the luminal membrane via the facilitated diffusion. Okay, so that is for protein products. Then for lipids, which are the fatty acids, the cholesterol, the monoglycerides, and whatever. Fatty acids, they diffuse across the membrane. Cholesterol is actively transported, that is across the luminal membranes now. Okay, now across the basal membrane, they get incorporated into chylomicrons inside the mucosal cells. The lipid products that enter into the enterocytes or mucosal cells, they get incorporated into chylomicrons, so they form lipoprotein, a lipoprotein that we call chylomicron inside the mucosal cells, and they enter into the lacteals. Lacteals are distal extension of lymphatic vessels. So lipids products do not enter into the blood, are not absorbed into the blood. They are absorbed into the lymphatic system. Alright, also note that most absorption take place in the proximal parts of the small intestine. Alright, then finally we look at the metabolism of these macromolecules. Now let's start with carbohydrates. Fate of absorbed glucose, that is from the blood. Now the absorbed glucose enters into the liver and is stored as glycogen. It enters into the muscle and is used for energy and glycogen synthesis. This same glucose also enters into the adipose tissue for fatty acids and fat synthesis. It also enters into the brain and other body cells for energy production via glycolysis. Alright, that's the metabolism of a rough overview of metabolism of glucose. Now for fate of absorbed amino acids. The amino acids, they enter into the liver and are used for protein synthesis or for gluconeogenesis. That is the formation of glucose from amino acids, or when they are excessive, they are broken down to urea, releasing ammonia. Okay, that is in the liver via urea cycle. Then they also enter into different body tissues for growth and repair function, for enzyme synthesis and hormone production. Okay, also the amino acids enter into the kidney and produce ammonia for ammonia buffer system in the kidney. All right, very important function of the kidney. Then finally, the fates of absorbed lipids. The absorbed lipids, which are the exogenous triglycerol, is stored in the adipose tissue and liver sometimes. The exogenous triglycerol is used as a source of energy in the liver, in the heart, and other body tissues. Free fatty acids are esterified with other lipids, such as free cholesterol, lysophospholipid, When free fatty acid is esterified with cholesterol, it becomes cholesterol ester. When it's esterified with lysophospholipid, it becomes phospholipids. Okay, or the free fatty acid can be used for energy production by the body cells. Okay, so that actually round off the whole section. I hope you enjoy it. Thank you very much for listening, and God bless you.